Welcome back, friends. We are talking about genetic mapping, especially the genetic mapping of eukaryotic system. Because it is much more complicated, we need to have special genes. We call them genetic markers. And then we correlate those markers with other genes and find those genes' position in the genome. Okay. So again, in the previous video, we have just talked about the introductory part of genetic mapping and the type of genetic mapping. And also we've talked a little bit about genetic markers and what are their functions. Now in this video we'll be talking about one kind of gene mapping, which is called the linkage mapping. So among gene mapping we'll be talking about linkage mapping. Okay. Now as the term suggests, linkage, I want you to focus on this. Linkage, it means whether two genes are linked or not. So this linkage map always put one question, which is whether two genes are linked or not. So two genes that we are dealing here, it could be two different genetic marker, or it could be one genetic marker, another gene of our interest, we don't know that. So these two genes are genetic markers. For example, here say G1 and G2. Now these two genetic markers, whether those two genes are linked with each other or not. Now here question comes, when we say whether two genes are linked or not, because these genes are present in a chromosome. Linked genes means the genes are placed pretty close to one another. They are pretty close to one another. On the other hand, unlinked genes are said that those two genes are distantly placed in the chromosome. Okay, so if two genes are pretty close to each other inside a chromosome, we call them a type of linked genes. Now here we will be seeing the importance of linkage map. Now I have told you before, the linkage map will give us a crude idea of whether two genes are placed closer or distantly. It will give us the position of one gene comparing it with another. Okay, so we cannot tell that this G1 is present in the chromosome number 2 or G2 is present in chromosome number 3 by this linkage mapping. We can only tell this chromosome, this G1 and G2, whether they are present in same chromosome or not, utilizing linkage map. So they are giving us the idea comparing the two position. Now the basic idea about this linkage mapping is that it can only be placed in case of the linear DNA. So if you are having linear chromosome, for example, say this is a linear chromosome, try to understand the concept very well. This is a linear chromosome. Somewhere here there is a G1. So let me give you two examples, not here. Let me give you two examples here. Say here, two examples, two different set of chromosome. Now we are taking markers, four different genetic markers. Here is say G1 and say the blue colored marker. Let me take it. This is G2. Now in this hand we are having G3, for example, and say G4. Okay, so we are having two different scenario, two different chromosome, and we are having two different set of genetic marker. Okay, in the first case G1 and G2, second case G3 and G4. So in this picture we know that where this G1 and G2 and G3 and G4 are placed, but in actual case when we need to find whether these two genes are placed in chromosomes, what are the distance between them, right? For doing that we utilize linkage map. So we suppose we don't know these facts. We don't know where G1 and G2 is sitting, where G3 and G4 are placed in the chromosome. How can we know this? Now what we do in this case, there are different progenies. Okay, now in F1 generation, if we are having these are the chromosomes. So what we do in case of plant, if we self those plants are put together for during the meiosis, and prophase 1 of the meiosis, these chromosomes which are aligned, the segment of the chromosomes are crossed over, right? Now here comes the importance of that place, crossing over. So if we place these things, say these are the markers, so we are having other, let me change the color totally. So this is the other chromosome. Okay, so these two are the different types, non-homologous chromosome, and the crossing over takes place between the non-homologous chromosomes. So here in this case, these two are the points, say. Okay, so this is the case, for example, say during the meiosis and prophase 1, segment of the chromosome are swapped with each other, called the crossing over. Okay, now if crossing over, so let me write the term crossing over. Crossing over, what do you mean by crossing over? Just segment of the chromosome will swap between each other, the random type of swapping. So it could cut from here, it can cut from here, here, or here, anyways, like that, right? For example, say one possibility of having a cut from this region, so what we'll have something like that. During the crossing over, we'll having, sorry, so we'll have this one at the end and this one at the end, then this, and then this. So these are the type of things that usually occurs in crossing over. Okay, or say rather this. So if it has a crossover from this side, then it will end up with something. So these different layers of crossing over can possible. Okay, different types of crossing over can possible, simply swapping of the genetic elements, simply swapping of the chromosome segments. But let's consider if the crossing over takes place between these G1 and G2, which are the selectable markers or genetic markers for our choice. We know the functionality of G1 and G2. If they are carried together, they give us a particular function. If they are not carried together, if only G1 transfers, they won't give us that function. If only G2 transfers, they won't give us the function. They only can give us the function if they travel together. We know that. Now suppose during the time course of crossing over, the break between the chromosome happens between this. It can possible because G1 and G2 are separated distantly apart, so it can have a break from here. If it is encountering a break from here, and then what it, sorry, not blue, so what it does as a result, say again, this kind of swapping event can happen, and then after that what it will produce, it will produce these two things. So yes, like that. So the first one is going to receive, so if this is the first one, this is the second one, first one is going to receive the blue dot, so G2, and this one is going to receive the red dot or G1. So utilizing the crossing over, what it can produce, the segment was changed or interchanged between these two chromosomes, and then they will have something like that. Okay, so if you are talking about the parental type, it should be G1 and G2 placed together. But this type of chromosomes will not be called as parental type chromosomes. Instead, this will be called the recombinant type chromosomes because they are recombined. The genetic segments are swapped. So these are recombinant type or denoted with small r. These are parental type are denoted with small p. Okay, similar things can also happen in the second case. Say in the second case they can also cut between here, so say it can encounter a cut, it can encounter a cut from here or here or here or in between here or something like that. So these are the random segment change between them. Crossing over is not specific, I'm telling you. It can cut from different regions and can swap segments between themselves, but it is rarer to cut between G3 and G4. Why? Because they are placed very close to each other. So by random chance, if we cut by leaving these distances, then G3 and G4, the probability of cutting in the middle of G3 and G4 are very less. Right or not? Right? But in case of G1 and G2, as they are placed distantly apart, the probability of getting a cut randomly will increase. So if we are having the distance between two markers greater, as we are getting greater, that is telling us the probability of having a random cut is more. But if we are decreasing the distance between two markers, then the probability of getting a cut, random cut, is less. That is where I am going to focus on. That's why I brought two different situations. So it is extremely rare to get this. So after the crossover also, in this case also the crossover happens and things will change. Say for example, in this case also say recombinant progenies are generated, but here the recombinant things, though the recombinant things generated, but these recombinant things will never disrupt the placement of G3 and G4. Remember, these are also recombinant type, but here the G3 and G4 are placed in the same chromosome. But in this case, as G1 and G2 are distantly related, distantly placed, there is a tendency of having a cut between them and being separated into two different chromosomes. So in the first case, if the trait, if the expression of the markers is in such a way that G1 and G2, if they express together, we get the phenotype. Now in this case, we won't get the phenotypic expression. By assaying the expression, we can tell there is a problem between, there is a cut between G1 and G2 by testing those, by testing it biochemically or molecular, with the help of molecular techniques we can tell it. But in this case we cannot tell, because whether the recombination happens but this G3 and G4 remains together. But the question arises, can this G3 and G4 separated? Yes, they can separate, but the probability of separation is very low. Okay, so due to this random crossing over and swapping of genetic segment, we can have the change. Here comes the importance of the term linkage. Now we can tell this G3 and G4 genes are closely linked with each other than G1 and G2. G1 and G2 is not that much closely linked. If they are, then we can get, they cannot be separated by recombination. It's a normal random chance recombination, crossing over, which separates two things apart. Right? Now we can't, now an important thing, we don't know whether the markers are placed closer or distantly, but by getting the expression. Now if we look at the expression from these things, if we look at the expression can be measured. Remember, three different type of thing can be, expression can be measured visibly, or it can be measured biochemically, or it can be measured molecular, with the help of molecular techniques. So we'll measure this expression, and by measuring this expression we can tell that this G1 and G2, they are separated, so they are probably are having less distance than this G3 and G4, just by looking at this data. But actually utilizing linkage map, we can tell the actual distance between these two different genes or genotypes. How? After this F1 generation cross, so this was the F1 generation, remember, this is the F1 generation cross. After that, what we get? The F2 progeny. In the F2 progeny, we can find the frequency of having this kind of chromosomes. The frequency of having this recombinant type and parental type. In both the generations, we are having parental type and recombinant type. In the F1, we are having all parental type because previously no crossing over has been made. But in F2 generation, we are having recombinant type. Not only recombinant type, but also we must have parental type because all the chromosomes are not crossed over. This is also another fact. All the chromosomes are not crossed over. So we'll be having recombinant type as well as parental type chromosomes. Okay. So this recombinant frequency and parental frequencies will be known to us after this F2 generation. And by calculating the frequency of parental type and recombinant type in this F2 generation, we can actually get what is called the recombination or linkage frequency or the recombination frequency. Now, what is the formula for getting the recombination frequency? We call it, the formula is simply like that. The number of frequency, so recombination frequency will be on the top. So that number of recombinant chromosomes we are getting, that number of recombinant genotype we are getting, divided by the total number of parental phenotype in that generation plus the total number of recombinant frequency of that generation. So recombinant type among the total number of organisms, that is the parental type as well as recombinant type in the same generation. And if we are going to make it a percentage value, we must multiply it with 100. Okay, so here let me change the R with a different color. Yes. Okay, so this is recombinant frequency, P for parental phenotype. So suppose in a case, suppose for an example, say this frequency of parental phenotypes here, parental chromosomes are say 40, and the recombinant type is, so parental type, there are two different parental type there must be. So say one is 40, one is 35, 36, and the recombinant type, say one is 10, another one is 14. So the recombinant, 10 recombinant and 14 recombinant are coming, and the parental type frequencies are there. It's just arbitrary values I am taking. Parental phenotypes, there are parental chromosomes that, for example, there's a 40 and 36 because we are having two sets, so one for 40, one for 36, and recombinant type developed in 10 and 14. So then for the same data for G1 and G2 markers, what we can get is 24 divided by 76 plus 24. So this is 24, this will be 100, so the answer is 24%. So the frequency or the recombinant frequency here is 24%. That means what this recombination frequency actually mean to us. Remember, apply the basic knowledge. Students often find this thing confusing, but apply the basic knowledge. If two genes are closely placed, then the chance of getting separated by crossing over is less. If two genes are distantly placed, the chance of being separated by a crossover is more. So if the frequency of generation of the recombinant type is less, that means they are much more closely related. So this recombination frequency that we are getting, this is actually directly proportional to the distance of two markers, right? Because everything is related between the distance, everything is related between this distance between two markers, right? If they are close enough, there should be less recombination. If they are distant apart, there should be more recombinants. So if they are close enough, they are less unlinked, more linked. Now in this case what we are getting, the unlinked thing or the frequency for unlinking. Now if they are closely placed, they are less unlinked, more linked. If they are distantly placed, they are more unlinked, less linked. Okay, so this value of recombination frequency is directly proportional to the distance between two markers that we are taking. So 24% of recombination frequency, it is going to tell us that the genes, that this marker gene G1 and G2, they are 24 unit distance apart in chromosome. We don't know what the unit. We'll be knowing a little moment later. That means this G1 and G2 are placed 24 map unit distance with each other. So if we draw this gene chromosome, then then this G1 and G2, they are 24 map unit apart. Okay, now this map unit is termed as centimorgan. As simply the centimeter is termed centimorgan. And why it is called centimorgan? Because this total idea of this linkage and linkage mapping is developed by Thomas Hunt Morgan, and he's a great scientist who worked with this Mendelian inheritance and developed many non-Mendelian inheritance patterns and explained many non-Mendelian inheritance patterns utilizing mathematical theories and explanations. And he also developed this kind of linkage mapping system. So to give him privilege, we call them 24 centimorgan. M is in capital. Okay, now it is stating that these two genes are 24 centimorgan separated. If the recombination frequency, now remember, now let's look at one thing. If we increase this recombination value, if the number of recombinant type is getting increased, what it means? It means the increase in this distance, right? So if we develop more recombinant type, that means more recombination are taking place. So if the value of this R is getting higher, that is meaning that more recombinant types are generated, so more crossovers and more recombinations are taking place. So this value will also get increased. So that will tell us those two genes are distantly related, right? And if the value of R is getting down, that means less and less recombination are taking place. So this value will also go down. That is going to tell us that those two genes are much more closely related. Okay, so they are directly proportional, directly linked. Okay, so this is the basic idea of linkage. And by using linkage mapping, we can tell whether two markers are placed with each other or not, and what are the distance between two markers inside the chromosome. Okay, and another important thing is that as this linkage map is dealing with the transfer of two genes, simply the transfer of two genes, whether they are together transferred or not transferred together. They could be transferred together, they can be transferred singly. During the production of gamete, if two genes are separated distantly or they are placed distantly, the probability of segregating them is more, right? Probability of segregation into two different gametes will be more. But if two genes are relatively closely placed, then the probability of segregating together will be more. Probability of segregating single would be less. Okay, so this procedure is dealing with the relative movement of genes and relative distance of genes and relative movement of genes in inheritance pattern. So this is also linked with the heritability of these genes or heritability of the marker genes that we are using. So in this case of linkage mapping, we have to use pedigrees. We need to use pedigree and we need to analyze pedigrees to learn whether a particular marker, our selected marker, is carried throughout the history of a family. Pedigree is simply the genetic history of a family. It is also can tell us different markers. If a disease we can find inside the family, we can take the marker for the disease gene and we can track whether from the bottom to the top that who is carrying those genes and how the genes is transferred from one generation to another generation to find out whether the genes are closely linked or distantly linked. Okay, but the pedigree analysis is also depend upon many different factors. We need to be very careful by using pedigree analysis, but this linkage mapping requires pedigree analysis. Okay, if you need to know more about pedigree analysis, you can find videos in my website, in my YouTube channel about pedigree analysis. You can go and look for them. So I'm not going to talk about pedigree analysis in detail now, but pedigree analysis is an important part of this linkage mapping. Okay, so this is the basic process of linkage mapping by getting the value of recombination frequency, and from the recombination frequency calculation what are the distance between two genetic markers. Okay, so that's it, and I hope this will help you. Thank you.